Hi, welcome back. A bit of a rainier week this week, but we're going to move on regardless anyway. So our topic today is transport across membranes. So to start off with, we really got to talk about where we would find membranes, their structure, the components of those membranes, and what each of those different components does and how it helps for the cell membranes to function. So when you're referring to cell membranes, ideally you want to be as specific as possible. Often I find that in the mark schemes in exam papers, they want specifically to say cell surface membrane rather than just cell membrane, because membranes are also located around other membrane-bound organelles, like the nucleus, the envelope around it has got a membrane, mitochondria have several membranes, etc. In general speaking, with this topic, we're focusing primarily on the cell surface membrane, so refer to it that. You can also refer to it as a plasma membrane as well if you wish, but I tend to go with cell surface membrane. So the cell surface membrane are actually surprisingly complex. They're only about 7 nanometers in thickness, but they are made up of many, many components. We've never really been able to image them properly because they are so thin, but based on the experiments that have been done and isolating the various components of them, a model has been developed to explain how they work, and this model was referred to as the fluid mosaic model. Fluid because all the components in there, primarily the phospholipids, are free to move within that membrane. They are free to move around, so it is fluid in that respect. A mosaic because it looks a little bit like a Roman mosaic, like little tiles put together, because it's like a sea of these phospholipids with a wide variety of different proteins embedded within this membrane, but again they are free to move as well. So that's what the cell surface membrane that is referred to as this sort of fluid mosaic model for in terms of how it works. So what are some of the key components of the of cell surface membranes? Well, primarily the most important part is that phospholipid bilayer. So 2 layers of phospholipids with their phosphate hydrophilic head sticking outwards, and then within the interior of them are those hydrophobic tails. So they collectively form that phospholipid bilayer, and that makes up the majority of the cell surface membrane, and acts somewhat as a sort of selective barrier. It will allow some molecules to move quite happily, simply diffuse across, whereas other molecules cannot. What else is in there? Well, there are, like I said before earlier, when I was mentioning the fluid mosaic model, there are a multitude of different types of proteins in there. So we've got channel proteins, which are proteins that extend all the way through that phospholipid bilayer, and they have a channel, a tube, that enables particularly charged molecules like ions to quite freely move between the interior and exterior of the cell, enabling them to cross the cell surface membrane, because they would normally be repelled by the phospholipid bilayer. So that allows facilitated diffusion, which is one of our key bits of our topic we'll talk about later. We also have carrier proteins. Again, these extend all the way through the membrane, and they allow the kind of one-way flow of molecules. Now, some carrier proteins allow passive movement, facilitated diffusion, whilst others require the use of ATP and will actively transport molecules and ions into and out of the cell. So there are 2 main types of proteins. There are a wide variety of others as well. In addition to those proteins, we also have glycolipids and glycoproteins. So I'll start with glycolipids. Glycolipids will superficially look like a phospholipid, but they will have a carbohydrate group sticking out of the top of that sort of phosphate head. Alongside them, we also might find these glycoproteins, and these are where we've got a protein molecule sticking through the membrane, and it again has been modified. It's got that carbohydrate group on the surface. So glycolipids and glycoproteins have a wide variety of roles within cell surface membranes. They're used as sort of recognition or binding sites, so for things like hormones or white blood cells can recognize and bind to them. They can also be used for cell-to-cell linkage and things like that. So quite a wide variety of roles, those glycolipids and glycoproteins. The other component of cell surface membranes is cholesterol. So cholesterol is a lipid. we hear a lot of bad press about cholesterol, but it is essential within our bodies, and in particular one of the roles it plays within our cell surface membranes. And what you'll find is that cholesterol molecules can be embedded within those hydrophobic tails inside within that phospholipid bilayer. And what the role of cholesterol is there is it reduces the fluidity of the membrane, which will make it less permeable. And you'll find that organisms that live in hotter environments, like sort of hydrothermal vent bacteria and things like that, will have a higher proportion of cholesterol to reduce that fluidity of their membranes in those hot environments, because it's got all that kinetic energy and their phospholipids will be moving around too much, and thus making them very permeable. The cell membranes might even cell surface membranes might even start to rupture. So that cholesterol will greatly reduce that fluidity. Whereas if you live in a very cold environment, something like a cod or something living maybe at 4 degrees C down in the North Sea somewhere, it will probably have a reduced amount of cholesterol within its cell surface membranes to increase the fluidity, to make its membranes more permeable at those low temperatures. So that's a nice sort of overview of the structure of our cell surface membranes and the components there. Other things to note are sort of factors that might affect permeability. So temperature, things like solvents like ethanol as well can also increase the permeability of the membrane, and that kind of comes into play with one of the required practicals, where you may look at beetroot and betulin being effectively released from within the vacuole of beetroot cells, and that again all is affected by the permeability. So you might either use temperature or something like ethanol as your independent variable there. So let's move on to the next phase of this topic, very, very important, and that's the ways in which materials are actually transported across the membranes. And those processes are simple diffusion, facilitated diffusion, osmosis, active transport, and co-transport. Okay, so let's look at diffusion. So like I said before, there are two types of diffusion: simple diffusion and facilitated diffusion. So what I've drawn here is a very simplistic view of part of the cell surface membrane. We've got that clear phospholipid bilayer extending that way, and here I've drawn a channel protein. So some molecules, like oxygen and carbon dioxide, can quite easily diffuse across that cell surface membrane because they're small, they're not charged, they're not polar. Or other molecules that are lipid soluble can quite easily simply diffuse across the phospholipid bilayer, no issue at all. Other molecules or ions might struggle to get across. Maybe they're too large, they're charged, or they're polar, or what have you. They cannot readily diffuse across that cell surface membrane, so they require some help. And that help comes in the form of those proteins I mentioned earlier, and in this case a channel protein, but also carrier proteins would be appropriate here. And they facilitate that diffusion, they allow it to become possible. So charged ions would normally be repelled by that cell surface membrane. This channel provides them a route for them to actually pass between the interior and the exterior of the cell. So that's facilitated diffusion, so bear that in mind. Look out for it. Both might superficially look similar in terms of data. You would expect potentially, initially a very high rate of diffusion if the gradient is very steep. As the gradient becomes shallower, i.e. now that there's less difference between the concentration inside and outside of the cell, you would eventually start to see that begin to level off and eventually plateau, and that plateauing area is where we reach equilibrium. And again, I should have probably mentioned this at the beginning, the definition of diffusion is basically the movement of particles from an area of high concentration to an area of low concentration. It's better to word it at A level, so the movement of particles down their concentration gradient. I would prefer to see that, but to help link it with GCSE, that's what you would say there. So that's that. Another thing to remember here as well, you're only going to have a finite number of these channel proteins within your cell surface membranes. So even though I might potentially have a very high concentration outside and a lower concentration inside, i.e. a very steep concentration gradient, there will be a limitation to the amount of uptake I can get because I've only got a certain number of channels. So a bit like, if you're going to a supermarket checkout or what have you, you'll be limited by the number of checkouts available. You might potentially have lots and lots of customers trying to get through, but you're always going to be limited by the number of checkouts. In this case, facilitated diffusion will be limited. It's a limiting factor by the number of carrier proteins or channel proteins there. So bear that one in mind. So that's a nice little overview of simple diffusion and facilitated diffusion. Let's move on to our next one, which is osmosis. Cheeky little tea break here, just going to affect the osmotic potential of my blood. Lovely jubbly bit of osmosis. Right then, osmosis then. So this one is a special kind of diffusion. It's the diffusion of water molecules. But it is more complicated because we're not dealing with just water. It's affected by other solutes in solution. So we've really got to understand and have a really good understanding of what a solution is. So a solution is where you've got a solvent, in this case it would be water, and a solute. And that makes a special kind of a mixture that we call a solution. Okay? So like I said, our solvent in this case is water, but our solute might well be something like sucrose, or it might be mineral ions, whatever that may be. So that makes things a little bit more complicated, because when we're referring to concentration, are you talking about the concentration of the water, or are you talking about the concentration of the solute? In general, I prefer to refer to the actual solution, that mixture of the two. So in order for osmosis to occur, we also need another structure to be in place, which obviously links with this topic, and that is that cell surface membrane. And that acts as a barrier. It will prevent the solute moving across it, but the water will be able to freely move between either side. And that membrane, therefore, kind of sets up this potential for osmosis. The water molecules are free to move across, but the solutes aren't. And therefore, we can get this kind of wholesale movement of water, and it can exert quite enormous amounts of pressure. I've even done it in experiments. You can actually get it to lift up the water level through osmosis. It's quite impressive. So let's look at some key things here. And one of the key kind of terminology we use at A-level is this term of water potential. So the highest possible water potential you could have is 0, and this is the symbol for water potential. I think it looks a bit like Poseidon's trident, which I find quite kind of helps me remember it because it's linked with water. So that's the maximum water potential you could have. This is basically distilled water, pure H2O, no solute dissolved in there. As I start adding more and more solvent, that value becomes increasingly more negative. So let's look at just some simple examples. So maybe I've added a little bit of salt here, I start adding a little bit more, and so on, and it becomes increasingly more negative. The more negative it is, the less water there is and the more solute there is. Because water is a universal solvent, or largely, a very strong solvent, and it means any kind of mineral ions or sucrose and things, the water molecules will be attracted to them, and that's what actually brings them into solution, and that means that there are less free water molecules to move. So in this situation here, I might have started off potentially with the same volume of water, but because I've added more solute, there's less water molecules free to move. They're now kind of attracted to those ions, and therefore they can't cross the barrier. So the water is always going to want to move from an area of high water potential to an area of low water potential. So let's look at a couple examples. So let's just say you've decided, although sadly you can't do this at the moment because McDonald's is closed, which is bitterly disappointing for me. I'm really looking forward to my Big Macs again. But let's say, when all things get back to normal, I can smash a Big Mac again. Go for it. Going to go for a large Big Mac meal. Going to have the fries. And what the hell, I'm going to have regular Coke as well. Really push the boat out. A bit naughty, but I'm going to do it. So you've eaten that meal. You've then started to digest it. And obviously the mineral ions present, the salts present in the chips, and the sucrose present in the drink. The sucrose, of course, will eventually get broken down into glucose and fructose. They will be absorbed into the bloodstream, and what's going to happen there is they're going to lower the water potential of your blood quite significantly. So let's imagine this is a scenario. So here I'm going to draw a blood vessel, a capillary. These are the cells, the endothelium lining the capillary walls. So there's the nuclei in there. Here's the blood plasma, and let's have some red blood cells in there as well. So I've just smashed this Big Mac, large fries and a Coke, and as a result, my water potential of my blood, my plasma, has dropped because of all those solutes. Let's make up a value. Let's say it's, negative 40 water potential. Your cells are haven't got pure water, and they've got their own salts and sugars dissolved in them, but they're going to be proportionally have a higher water potential. So let's say they're 20. I'm not going to do all of them, but you can imagine all the cells on this diagram have got a negative 20 water potential. So what would happen in this scenario is that I've got a lower water potential in my blood plasma and a higher water potential in my cells. So the water, therefore, would want to move from an area of higher water potential to an area of lower water potential across that semi-permeable membrane via osmosis. That's how I would word it every single time. So the water would start to leave all of my cells, those endothelium cells, tissue surrounding it, the red blood cells, into my blood plasma. So all these cells here would become plasmalized, they'd get kind of floppy. And the volume of water inside my plasma would go up, therefore my blood pressure would shoot up too. So if you were to do this regularly, and you maybe have got some underlying health problems, having those sort of salty, sugary foods can have quite a dramatic negative effect on your health by increasing that blood pressure. So in this scenario, with osmosis, I'm going to assume that my cells have the same water potential as they did before, but I'm going to start doing something that might not seem like a big deal. I'm going to drink loads of distilled water, pure water. Surely drinking pure water will be fine, that's not going to do them any harm. Well, actually, it will do. It will be quite catastrophic, the effect of drinking lots of distilled water. So what would happen to my blood plasma if I started drinking distilled water in certain quantities is that my water potential in my blood would start to increase. And let's say it got as high as minus 10 water potential, like that. Now, in this scenario, remember, we've got to remember osmosis, water will always move from an area of high water potential to an area of lower water potential. So if my blood plasma is now higher water potential, water will start to move from the plasma into the surrounding cells. So what would happen in this scenario? Well, because these are animal cells in our bodies, those cells would eventually burst because there's no cell wall to provide any kind of mechanical support. So all that water would move into our cells, causing our red blood cells, endothelial cells, and any kind of surrounding tissues to actually burst. So this would be quite catastrophic if you were to drink distilled water. So believe it or not, tap water is not pure water. It has got mineral ions dissolved in there. Not as much as seawater, but it has got a more negative water potential compared to distilled water. So there's two kind of examples there. I did sort of allude to a little bit of difference between animal and plant cells when it comes to osmosis. If they're in a solution that has a higher water potential than they do, that in plant cells, their cell walls can actually resist that kind of inward movement of water by osmosis. They won't burst, they'll just become turgid. In fact, plant cells ideally would prefer that, gives them extra kind of rigidity. Whereas animal cells, because we've got no cell wall around our cells, they will just simply burst. So there's a big difference there. So let's move on to our next key process, and that is active transport. So compared to the processes we've seen so far, this is the first one that actually requires the input of energy, hence the name active. That energy comes from the form of ATP. And ATP, of course, is generated during aerobic respiration in our mitochondria. So I think of active transport as a bit like greedy transport. I've got a lot already, but I want more. I've got a lot already, but I want more, right? So we're moving against the concentration gradient. So in this case, I've got a higher concentration within my cell and a lower concentration outside of my cell, but I still want more of it. And therefore, I've got to move it against that concentration gradient. In theory, those, in this case, ions, want to diffuse that way. I'm not going to allow them, but I want to try and get these ones into my cell just to maximise the amount of uptake as possible. So how does active transport work? Well, first of all, in addition to needing ATP, we also require these carrier proteins. So these carrier proteins are a little bit like a one-way system. I think about those sort of one-way doors. You can go through one way, but you can't go back the other. You know, you're blocked that way. In order to open and close them, to basically pump these molecules in, we require the use of ATP. So an ATP molecule would come along, it would be hydrolyzed, and the phosphate donated to it would actually cause the shape of that carrier protein to change. So if I just quickly sketch what that would look like that, and that is actually enabled, there's the phosphate there, enable those molecules to be moved in against a concentration gradient and prevent any other ones we've already got from escaping. So that's active transport. Key things to note with this one, you might see it linked with cells and organelles, and key organelles that might be linked with cells that are doing lots of active transport, of course, they're going to have more mitochondria than normal to generate more ATP from respiration. You might even get the link between ribosomes and protein synthesis to actually make these carrier proteins. So you can see how this topic kind of overlaps quite nicely with that of the cells topic. So we're going to look at another aspect of active transport, well active transport's involved with our final exchange process, which is called co-transport. Right, co-transport then. So co-transport is probably the more complex transport processes across cell membranes. And it involves a couple of key steps. I'll break it down. So what I've drawn here, or at least crudely drawn, is an epithelial cell in the small intestines. Here I've drawn a capillary. So you can imagine this would be part of maybe a villus within the small intestine. So here's the lumen, here's all my food molecules like glucose. I've also got a high concentration of sodium ions. They've been added further up in the digestive system. And I'm trying to transport that glucose into my blood. Now, first, the glucose will happily diffuse into the cell by facilitated diffusion via these channel proteins here. But as the concentration of glucose starts to decrease in the lumen and starts to increase in the blood here, you will see that it might reach equilibrium. So effectively, no more diffusion is going to take place. And if we left it at that, you would lose a big chunk of that glucose and other important nutrients like amino acids. They would simply pass their way through the digestive system and be released when you went to the toilet. Not an ideal situation. And again, this is that kind of idea of that greedy transport. I've already got a lot already, but I want more. So in order to maximize the uptake of molecules like glucose, we do something called co-transport. And it does involve active transport. Now, the first thing that happens is down here. And I've got a specialized protein here called a sodium-potassium pump. And what that does is it requires ATP to operate. And what in this case it does, it will pump potassium ions into this epithelial cell, but it will pump the sodium ions out. And actually the pumping of the sodium ions from the epithelial cell into the blood, and always write that, actively transport sodium ions from the epithelial cell to the blood is the most important step here. I'm not too worried about the potassium ions at the moment. And what that does is it creates a very steep concentration gradient between the lumen and the blood. So I've got a high concentration of those sodium ions in here with all my food molecules, and I've got a low concentration inside my cell because I've been pumping them out, actively transporting them out. So the sodium ions will start to flood back into the cell via facilitated diffusion, going down their concentration gradient. And as they're flooding back into the cell, they're going to start bringing in glucose along with it. A bit like, imagine you're sort of minding your own business, walking along, and suddenly a huge crowd of people storm into you. You're going to be swept along, you're going to be pushed away with the crowd, aren't you? And that's what's going on there. So I'll break that down again. So number one, sodium ions are actively transported from the epithelial cell into the blood. This creates a steep concentration gradient between the epithelial cell and the lumen. So the sodium ions will diffuse into the cell via facilitated diffusion. This will bring the glucose in with it against its concentration gradient. Once the glucose is inside the cell, it can then be transported into the blood by other carrier or channel proteins via facilitated diffusion there. So that's how co-transport works. So a complicated one, usually worth 3 or 4 marks, but try and word it in the same way that I do. You might come across these sodium potassium pumps as well when you look at the nervous system in year 13 as well. So they have a very important role there in generating action potentials and so forth. So that's it's quite a small topic, this one, but quite information dense. So hopefully you found that all okay and interesting. So this is Dr. P signing off on this wet day. You stay safe, you stay happy.